have to tell everybody.
John couldn't make it today, so that's why I'm introducing Michaek Levine is going to talk about complementary frame reconstruction. And he's a grad student at the Martinos Center from the Harvard Physics Department. Biophysics Department. So, before you.
Thank you. Thank you everyone for coming to Science on Tap today. Like Micah said, I'll be talking about complementary frame reconstruction for PET imaging. So most of the talk is going to be devoted to describing how PET reconstruction works, for those of you who don't already know. And then I'll get to complementary frame reconstruction at the end and show you some of the results I have. So when I think about acquiring and reconstructing PET data, I tend to break it into three parts. First, the acquisition phase, where we put the patient in the scanner, inject the radioactive tracer, and then acquire the data. Next, we use that acquired data to do the reconstruction, and then finally, for quantitation, we take the reconstructed data and use it all to derive parameters that are of physiological interest, which vary by radiotracer, depending on the molecular targets. Right? And so today, I'm going to be talking about the acquisition and reconstruction parts of that pipeline. So, these slides, this slide was first made by Daniel Chande, who's actually in the audience today. I'm glad to see him here. So this is the nucleus of the radioisotope that has been injected into either our patient or our phantom, and because it's radioactive, it tends to decay, emitting a positron. This positron is a Bernoulli kinetic energy, moves around a little bit until it meets an electron, at which point an annihilation event happens, and it sends two gamma ray photons in opposite directions, which we can then pick up with our photodetectors that form a ring around the outside of the PET scanner. And this is what that looks like. The annihilation can happen anywhere within the PET field of view. Right? And although we know that the photons get sent in opposite directions, they could hit any two pairs of detectors anywhere in the scanner. Right? And so we need to be able to account for that. Now, if we get a coincidence event with the pair of photodetectors, right? We know that the annihilation event came from somewhere along this line, but we don't necessarily know where. This is called a line of response. Right? So in order to reconstruct the data, we take this line of response data, this long stream of events, and we rearrange it into something called the Sinogram. And the way that we do that is we look at all of the LORs at a given projection angle. Right? And we form the first row of our Sinogram by looking at all of the LORs at that angle. Right? And so what we do is we effectively take a projection, right, in which those intensity values correspond to how much activity was observed along all of those lines of response. And then we move that to the first line of our Sinogram. Then we iterate the projection angle and do the same thing again, where the columns correspond to each LOR at that angle. We take a projection, and we move it to the next line of the Sinogram. And we continue this until we've covered the entire space. And effectively we have a different representation of our data. So, but in reality, a Sinogram doesn't look exactly like this. This is a bit of a cartoon. A Sinogram actually looks like this. Right? And you can see as you move downward, that is, as you iterate the projection angles, you can see the data takes a bit of a sinusoidal form, hence the name Sinogram. But for the purpose of explaining how Sinograms work, and for the purpose of explaining how iterative reconstruction works, it's easier to use the cartoon version. Right? So, for iterative reconstruction, I've been operating as though we know what our activity distribution is. So that's a phantom right there, right? But before we've reconstructed it, we don't necessarily know what it looks like, right? All we have is the LOR data, the Sinogram data that we've acquired from the scanner. So what we do instead is we start with an assumption. An assumption, say, of uniform activity throughout the entire phantom. And we can do the same thing, where we project along all of our different angles, right, and form the same kind of sinogram as we did for the acquired data, except because this is a uniform phantom, we'll get a fairly uniform looking sinogram. Right. And so from this, we can estimate how accurate we can look at how accurate our estimate is relative to the data that we've actually acquired. Right? And this gives us a cost function. It tells us how wrong our guess is and how much we need to update it. But there's one more thing that we're missing, because the cost function is now in projection space, in sinogram space. But we need to update our estimate of the activity in image space. Right? So the one thing we're missing is a system matrix. And the system matrix, along its columns, has all the projection bins, so every single pixel in the sinogram. And in its columns, it has voxels, so every single voxel in the image, which means that the system matrix is enormous. Right? But effectively, it's a mapping of all of the voxels in your image to all of your projection bins, because in principle, activity in a given voxel could end up in any or all of your projection bins. So the system matrix is based on the geometry of the scanner, and it helps tell you how image space and projection space are related. So what we can imagine doing is taking the cost function, running it through the system matrix, and giving us an updated activity estimate. And then we can do the same, we can do the projection the same way I've been talking about, to get a new sinogram estimate. So this was our first iteration, everything's white. But if we do, say, 10 iterations, we might get an activity distribution that looks a little more like this. Right? And when we project that, we can get a sinogram that looks a lot closer to the data that we actually measured from the scanner. And then eventually, by say iteration 50, we'll converge on what the actual activity distribution of this phantom is, and the sonograms, our estimated sinogram, will match our acquired data. So that's how reconstruction works in general. So next I'm going to talk about complementary frame reconstruction. But first, there's one more thing I need to cover, and that's that the reconstruction I just went through is for one single PET volume. Right? But with, say, an 85-minute scan, we can break up our PET data any way we want to. We could reconstruct the entire 85-minute scan, or we could take a smaller frame, say a 60-second frame. This is raclopride, which is a very specific tracer uptaken mostly in the striatum. As you can see, there's not a whole lot of activity outside of the striatum, which are those two bright spots. Right? But at 60 seconds, in a shorter frame, there's more noise, and at that point in the scan, there's more activity in areas outside of the striatum. Right? And this still looks all right, but what we can also do is we can take an even shorter frame. Let's say we take a 2-second frame, and that really starts to push the temporal resolution of PET acquisition, right? Which looks like this, where you can't really even make out the striatum amongst all the noise. And this 2-second frame is exactly where complementary frame reconstruction can help us the most, where the noise is the highest and where we want the temporal resolution to be the lowest. Temporal resolution should be the highest, the smallest temporal resolution. So this is how complementary frame reconstruction works. We can imagine our PET data. Let's say we're interested in a 1-second frame of interest, and we have 60 seconds around that. So we can also think of a 59-second difference frame, which represents basically just the 60 seconds with the 1-second frame of interest taken out, like this, in this mode form. We can turn these into sinograms, doing projection the same way that we did earlier. And if we want to look at the 1-second reconstruction, we can directly reconstruct that short frame sinogram, right, which looks like this. Call it IFR independent frame reconstruction as opposed to complementary frame reconstruction. But what we can also do is we can take the long frame sinogram and difference sinogram and reconstruct each of those. And then we have a long frame image and a difference image, and if we subtract the difference image from the long frame image, we get a complementary frame reconstructed image that's of the same duration as the short frame image directly reconstructed. But you can see, by looking at it, the independent frame reconstruction is noisier than the complementary frame reconstruction. And this is all based on work that was done by Inky Hong at Siemens, published in 2014. And he helped me implement complementary frame reconstruction here at the Martinos Center on some of our data from the MMR, which I'll show you now. So on the top, this is rapid pride data again. So it binds in the striatum to dopamine receptors. So up top we have independent frame reconstruction. On the bottom we have complementary frame reconstruction. And you can see, you can resolve a lot, you can see a lot more in the complementary frame reconstructed images. You can actually see the striatum much better. So this is without any smoothing. This is a 4 millimeter smoothing kernel, and this is an 8 millimeter smoothing kernel. So that's pretty nice. So these are some preliminary results. So what's next for this project is to look for the applications where complementary frame reconstruction can help us out in this, and look at the parameters, right? Ask how much can we push that temporal resolution? Is it 2 seconds? Is it 1 second? Is it less? And what size should the window, the complementary window be? Should it be 60 seconds or 30? Does it need to be longer? Things like that. So I'd like to thank Chipper and Katana, my advisor, particularly Inky Hong and Siemens, who developed this method. Dan Shonde, who's here, who did some of this work a long time ago. I picked it up now. Jim and Michelle at Biophysics, my thesis committee, everyone in the lab, and thank you. So, we don't take questions here at Science on Tap, but if you want to know any more about PET reconstruction, come talk to me. Thank you.